So I study cardiac glycosides, which are a class of specialized metabolites that function by inhibiting the sodium-potassium ATPase pumps in animal cells. And I'm interested in how they're synthesized. So cardiac glycosides have evolved independently in 12 different plant families. And so this is like a great example of convergent evolution. And so in that sense, they're ecologically important. And they're also important medicinally because they're used as heart treatments for things like arrhythmia or congestive heart failure. So there's some interest in how they're synthesized for that reason as well. I'm looking specifically at cardiac glycosides produced in tropical milkweed, milkweed being an Eposinaceae, one of the families that produces cardiac glycosides. And I'm looking specifically at a molecule called verucherin because it has sulfur and nitrogen, which is a little bit unique compared to most cardiac glycosides that don't have sulfur and nitrogen. So in all of the 12 plant families that produce cardiac glycosides, there's no like pathway that's been fully elucidated. So we're sort of like taking stabs in the dark a little bit. And so we thought it'd be easier to start with a more unique cardiac glycoside because it'll probably be easier to find a gene that performs the sulfur and nitrogen incorporation than trying to find something that like produces like the base structure of a cardiac glycoside, because it's like all very general. There's like a steroid core, lactone ring, sugar groups, like really anything can kind of build that. And so some of the information that I had going into my project is that we know that verucherin is induced by jasmonic acid, as with other cardiac glycosides. So basically an experiment was done where milkweed was treated with jasmonic acid, and then we saw increased expression of verucherin after that. So we have an idea that we're looking for genes that are going to be involved in jasmonate signaling pathways. A few years back, a paper hypothesized that the sulfur and nitrogen in verucherin comes from the amino acid cysteine. And so we also did experiments with stable isotope labeled cysteine, where the plants were treated with that. And we saw that plants that didn't have the isotope labeled cysteine compared to ones that did, there was increased expression of that like heavier verucherin. And so that also gave us the idea that we should be looking for a gene that can perform a cysteine incorporation step, since that's very likely where the sulfur and nitrogen are coming from. And so what I'm doing specifically is comparing genes that are differentially expressed under stress in tropical milkweed, Asclepias curassavica, compared to incarnata, which is a closely related species that does not produce any sulfur and nitrogen cardiac glycosides. And essentially, the idea there is that both of these species will be producing, will be on defense and upregulating certain genes, but none of the genes that they're upregulating that they have in common will be those that are performing the sulfur and nitrogen incorporation step, since incarnata isn't capable of doing that sulfur and nitrogen incorporation step. And then there's sort of a similar relationship between Asclepias syriaca, which is common milkweed, and tuberosa, which is butterfly weed, where syriaca has the sulfur and nitrogen cardiac glycosides and tuberosa does not. And we think that curassavica and syriaca will probably be using similar genes to perform the sulfur and nitrogen incorporation step. And so that would be like further confirmation of any candidate genes that we might have if there's homology between those two species, because then it's more likely that it would actually be a gene that we're interested in. Some other information that I have available but can't exactly access yet, which I will explain, is genes that are strongly positively correlated with verucherin abundance. But the reason why I can't use that right now is because that was done a few years back, and it was using like an older version of the curassavica genome, so the gene IDs don't exactly match up to what I have. So I'm gonna go back and try and redo that to compare some of my candidate genes to the verucherin abundance data, because obviously if I have genes that are strongly correlated with verucherin abundance, those would be more interesting. But in terms of my actual candidate genes, I have like a short list here just based off of functional annotation, things that are involved in sulfur and nitrogen metabolism or potentially cardiac glycoside metabolism. And these are like some of the most highly expressed genes and significant in terms of p-value as well. And I'm going to compare that obviously to the data that I'd previously mentioned. But once I actually narrow down my list, there's two different approaches I want to take to testing those candidate genes. The first would be gene silencing with RNA interference. So creating a double-stranded RNA construct and interrupting the activity of those genes, seeing if I can change the cardiac glycoside profile and knocking out expression of cardiac glycosides, specifically fruition, of course. And then the other direction would be transient expression in Erythemum caranthoides, which is another species that produces cardiac glycosides. But someone in my lab had worked on that and created mutants that don't produce any cardiac glycosides. So I would express my genes in there to see if I could bring back production.
Thank you so much, Allison. This is super interesting. I'm really glad to have been able to hear all about your research. Thank you.